Assalamualaikum warahmatullahi wabarakatuh, and a very good day to everyone that watching the video. My name is Nur Kamilia binti Mohzani Aris. I'm from University Putra Malaysia. So this is my practical assessment tool, which I need to present a model of one of the bioremediation method that I've chosen. Before I dig down the details of the model that I've built, I would like to introduce some of the background of the model that I chose. One of the most common contaminants that needs bioremediation is hydrocarbons. This is because the threats that it poses to human health and also controlled waters. Hydrocarbon compounds derive from petroleum fuels, including the common that we know such as gasoline, kerosene, lubricating oils, and also greases. Even though hydrocarbons are organic compounds that contains only hydrogen and carbons, they include vast number of different other compounds, and each of that compounds have their own physical and chemical properties. Moving on to the remediation technique that I chose for my model, which is biopiles. So biopiles represent a reliable system that reduce the concentration of petroleum constituents from the excavated soils through biodegradation. So biopiling involves collecting and transferring contaminated soils to a site specifically set up for biopiling. This will stimulate the aerobic microbial activity within the soils through aeration and or addition of minerals, nutrients, and also moisture. And this leads to the basic component of biopiling, which are aeration, irrigation, nutrients and leachate collecting system, and also a treatment bed. So this particular ex-situ technique, which is biopiling, are often being considered because of its cost effectiveness, and also it allows the condition of the biodegradation being controlled, like the nutrient, the temperature, aeration, moisture are all being monitored while carrying out this technique. A little background details on the model that I've chosen. The type of contaminant is diesel fuel spills from the heavy machineries. The place is on a construction site. In the soil has exceeded the acceptable limit from the regulations. So since bioremediation is a common treatment used for remediating hydrocarbon polluted soils, it is suggested that biopiling is used. So why the need of remediating these contaminated sites? Since it exceeds the acceptable limits of diesel fuel in the soil, the groundwater below the construction site are also being tested, and it is found from a gas chromatograph readings that the groundwater also have high density of diesel oil. Since the area would be constructed and developed, so this problem needs to be solved before that even happens, because when the area are being built with buildings, and the groundwater will keep on being polluted by these contaminants. So moving on to the biodegrading microbes. The microbes are aerobic, hence the provision of the air for them. And also in this study, they do not mention specifically the species of the microbes. There are 297 bacterial isolates obtained. From my understanding, that they did not found which bacteria actually responsible to create these hydrocarbons, because most of the bacteria that are present at the beginning of the treatment are not found later, and vice versa for the bacteria that are not found on the beginning of the treatment and later found at the end of the treatment. The example that are given in the paper are there are two bacteria, which is Saccharopolyspora, which is found at the beginning and not at the end, and also Cochuria, which are found only after 18 months of treatment and not at the beginning. This biopile treatment took 540 days. The microbe used are the resident microbe, so they just enhance the microbial activity in the pulse by adding the aeration, moisture, and also nutrient. There are no new species introduced to the piles. And from the report, it says that although inoculant are added to the biopile, it doesn't introduce a new community, a new bacterial community to the biopile. The microbial activity also are not uniform throughout the biopiles. It chooses certain sites with preferable pressure, and at those sites are the most active microbial activity found. In this paper, what I found interesting is that the commercial inoculant are effective in lab, which artificial diesel fuel are introduced to them. But in real life, these inoculant are not really effective in introducing new community to the contaminated soil. For the byproduct from this treatment, from what I read, biopile usually produce few to none byproducts at all. This is because hydrocarbons are organic compounds, so the degradation of this compound produce carbon dioxide and water. Then the last one is do it need further treatment after the biopiling is completed? Not really, but the leachate needs to be collected and treated to avoid pollution from the leachate produced. So in the next part of this video, I will show you the model that I've built, and I will explain to you each component of the model and also explain what it is about. So what you're seeing on screen now is actually the whole look of what my model looked like. And as you can see, I divided the piles into two so that we can see the cross section of the model, so I can explain to you the other components that lie on the bed. Okay. So this is my model, and as you can see, there are 3 colored boxes: pink, blue, and also yellow. So the blue one here is the air injection pump, and then this is the leachate collection and treatment system. So this yellow one here are the nutrient and moisture addition pump. The next part that I want to explain is the physical structure of this model. So the first physical structure that is important is the lining or the membrane of the biopiling site. This is important because when the contaminated soils are excavated and were to put into the biopile site, it needs to be lined first. So why the lining or the membrane is important is because when the soil are excavated from the contaminated site, it is brought to the site to be biopiled, right? And then if the soil are not lined and the contaminated soil are just put there on bare soil, it might contaminate this site. So this lining is to prevent the migration of the contaminant into the soil. And also throughout the biopiling duration, moisture, nutrient, and maybe some inoculant will be added up to the pile. So we don't want all these substances contaminate the treatment site, where this should be a treatment, not further contamination in other sites. So the next physical structure for this model is the cover. This model uses a HDPE cover as the cover. Biopiles are usually covered to avoid the volatilization of the contaminants towards the atmosphere. Also, the cover helps to ensure the water content in the biopiles. The cover are also usually weighted with something heavy, so that when there is wind or anything, the cover won't be exposed or open. So for the next part of this video, I wanted to explain about the more technical components of this model. So the first thing that I want to explain is the air inlet. So this air inlet is located on top of the pile because in my opinions, for the air pump, it is located at the below surface of this pile. So to increase the effectiveness of aeration in the pile, they used an air inlet. The next thing is the soil vapor monitoring pole. So as you can see in this picture, the bamboo stick represent this soil vapor monitoring pole. So since in biopiling, the moisture, the aeration, the nutrient are all being monitored, so this pole helps in monitoring what's going on inside the pile, because in biopiles, the pile are not being mixed. Once it is put there after excavation, it stays until the biopiling complete. After that, I wanted to talk about the pumps. So basically there are 3 pumps: the air pumps, nutrient and moisture pumps, and also another one is not really a pump, but it's a leachate collection and treatment system. Like earlier in the video, I mentioned that before the excavated soil are piled up on the site, it needs to be lined first with the geotextile, and then after lining the base of the site, then the piping goes on. So the piping are for these pumps to function, because the pumps are only controlling what's going in and out, and so the pipes needs to be installed before the contaminated soils are put on site. For the nutrient and moisture addition pump, so this act two-way where the nutrient are added into the piles, also the moisture, which is the water, to add moisturization and also nutrient in the pile. And for the air, in the journal that I've referred, they did not mention about the air temperature, but in another journal, I found out that when they pump the air inside the piles, they also control the air temperature. So the temperature of the piles become more favorable to the microbiome. So for the leachate collection and treatment system, not all biopiling treatment need leachate system. It depends on the condition of the soil. So in my model, it includes the leachate collection and treatment system. So the leachate collection and treatment system is important so that the leachate doesn't overwhelm and goes out of the biopiles area and then contaminate the other area of the treatment site. Okay, actually I'm done explaining the model, what it is about, the function of each component. The next part is I want to talk about the limitations. The first and the most obvious limitations in biopiling is ununiform biodegradation. This is due lack of mixing when we are doing this biopiling method. As the name suggests, where it is piled. So the contaminated soil are piled in one place, and the pumps that I mentioned earlier do the job in providing air, providing moisture, nutrient. For the mobility of the microbiome to biodegrade, the pollutants are totally depends on them. Second limitation is the physical disturbance caused by the excavation and also the pile construction. In this process, the excavation also produce noise, dust, which will trigger other types of pollution in order to treat one type of contaminant. Third one is flat area is needed. So since biopiling are done in an open space, flat area is sometimes a trouble because the natural geographic of natural places are not usually flat, and flat area is needed for the installment of geotextile as the lining of the base, and also the installation of the piping system for the pumps that are used for biopiling. The next limitation is that climate condition influence largely on the treatment parameters. This is because the climate condition can interfere to the control condition of the biopile. Like for example, the moisture, the temperature. This condition needs to be controlled and maintained so that it is always favorable to the microbial activity, so that they don't stop doing their job and keeps the biodegradation rate at a healthy pace. The last limitation that I want to address is the volatilization of the VOCs must be addressed and monitored regularly. This is because biopiling are done in open space. We don't want these VOCs pollute the environment. So I think that is everything that I need to talk about my model. So before I end, I would like to tell why I choose biopiling. I think that biopiling are suitable for, like, from what I read, which is construction site, because they already have the site for treatment. It's just they need to prepare like specific place for this biopile treatment, and since they are constructing at that site, I assume that they can find that place easily. And also it produce less to none byproducts or pollutants. There are not really any post-treatment to the sites if the treatment followed strictly to the regulations, such as lining and also the leachate collection and treatment system. So it should be fine. After the treatment, the soil can be used again, and the site also can be used again.